Okay, so here we are, AP Bio, Chapter 7 on photosynthesis. We are now going to focus on the Calvin cycle or the light-independent reactions. Previously, we've covered a general overview of photosynthesis, and that we focused on the light reactions. So as we go through a quick review, here is the general equation for photosynthesis. Carbon dioxide and water using light to make glucose and oxygen. In our light-dependent reactions, we have used the water and we have generated our oxygen gas. So we talked about autotrophs, we talked about the structure of a chloroplast, we have talked about how photosynthesis is a redox process. We have talked about the energy carriers of ATP and NADPH and their role in connecting the light reactions to the Calvin cycle. We focused on the light reactions and how they use water as a way of generating oxygen. Now we're going to focus on using the ATP and the NADPH with carbon dioxide to ultimately produce our glucose or sugar molecule. We talked about how photosynthesis has these photosystems which gather light energy, use it as a way of transferring that light energy into the electron. The electron is then used to generate a concentration gradient so that we can then create energy in the form of ATP. We also talked about how the electron is ultimately being donated to NADPH as a way of producing that molecule. So now what we have done is we have made our energy molecules, and what we're going to do is use them in the Calvin cycle. The Calvin cycle is found in the chloroplast stroma, which is the area outside of the inner membrane and the area inside the kind of the structure or the external membrane. It consists of carbon fixation, which means we're going to be adding carbon to a molecule. We're going to be doing a reduction reaction. We're going to be releasing G3P, which ultimately is what turns into glucose. And then we're going to regenerate our initial molecule, RUBP, as a way of completing or connecting the cycle. So one of the big things is that there is two different types of photosynthesis that occurs. And I need you guys to understand that the two different systems are going to determine whether or not we can enter into the Calvin cycle. There is non-cyclic and cyclic. When we do non-cyclic electron transport, we're going to be producing the NADPH and the ATP with our byproduct of oxygen as a way of making the energy molecules we need for the Calvin cycle. However, if we do cyclic electron transportation, we're only producing ATP. Without the NADPH, we're unable to go through the Calvin cycle, and therefore we are unable to make sugar. Now, what happens in cyclic electron transportation is we're only going to produce ATP, and here's why. The excited electron that comes out of our photosystem number 1 is going to be passed to the electron transport chain. It's going to go from photosystem to photosystem and just continuously cycle as a way of generating or building up the hydrogen ion concentration, which is then potential energy that we use and harness to make the ATP. So chemiosmosis is still happening in cyclic electron transportation. Now, what's going on, again, is the electron is going from our P700 molecule back to our P700 molecule, just going round and round and round and round. And in that way, we only generate the electron gradient or the hydrogen ion gradient as a way of making more ATP. So if a cell needs ATP instead of glucose, it's going to do the cyclic electron transportation versus the non-cyclic that we have previously talked about. The non-cyclic is the one that you're familiar with. We go from photosystem 2 through the electron transport chain to photosystem 1, and then the electron is ultimately donated to NADP plus as a way of reducing it to make NADPH, the molecule that will need for Calvin cycle. If we don't need to go through the Calvin cycle, then what we're going to do is cyclic. With cyclic, we go from photosystem 1 back to the electron transport chain. Again, we're pumping those hydrogen ions, and so we can generate ATP, but what we're not doing is we're not donating the electron back to the NADP to make our NADPH so that we can enter the Calvin cycle. Okay, so now let's talk about the Calvin cycle. The Calvin cycle is going to take a carbon from the carbon dioxide, and it's going to fix it into a molecule. So if we were to look at the first part of the Calvin cycle, we're going to begin with a molecule called RUBP. RUBP combines with the carbon from CO2, and in doing so, we're going to generate two molecules of what are called 3-PGA. Now, the big thing to know about RUBP combining with carbon is that's an enzyme catalyzed reaction, and the enzyme that catalyzes it is called rubisco. Rubisco is one of the most numerous enzymes or abundant enzymes on the entire planet, and that's because this process is happening all the time. So we're starting with a molecule, RUBP, we're combining it with the carbon from CO2, and we're generating two molecules of 3-PGA. Here's where we're going to use the energy molecules that we've previously created from the light reactions, the ATP and the NADPH. And we're going to simply change or reconfigure the molecule from 3-PGA to a molecule called G3P. G3P is a molecule that has the ability to leave the Calvin cycle and go be converted into glucose or some of the other compounds that we might be needing in the cell. If G3P is not used directly to go make glucose, then it's recycled and it is turned into RUBP again using more ATP from the light reactions. So we can see how the Calvin cycle is unable to operate if it doesn't receive the energy input from ATP and NADPH, because we'd be unable to make G3P, and G3P is the product that ultimately goes to make glucose. Now, we call this process carbon fixation because in the initial reaction of the Calvin cycle, what we're doing is we're taking one carbon and we're adding it to a 5-carbon compound, our RUBP, which stands for ribulose 1,5-biphosphate. The enzyme that does that is rubisco, and it's the most abundant one in the world, and it's really important to kind of keep in mind that rubisco is super special in this overall process. Now, what are we doing? When we do our carbon fixation, the CO2 is combining with RUBP to make our 6-carbon compound, which initially gets broken down right away into 3-PGA. Once that happens, we're going to convert it into G3P using those energy molecules from the light reactions. And then that G3P can be used to make sugar, or it's going to regenerate using more ATP to regenerate our RUBP. Now, this is the general idea that what we've done, right? We've taken our CO2 and we've added it to RUBP in the Calvin cycle, and we've ultimately produced G3P, which is used to make a sugar. That sugar can go and be used to make cell walls, to go make starch, to go make cell respiration or turn into ATP through cell respiration. It can be used to make any other organic compound that cells need to function. However, this process of doing the Calvin cycle has been specialized by plants over time. So what happens is in a C3 plant, if there's a drop in the concentration of CO2 and there's a rise in oxygen, what happens is that they can divert Calvin cycle to photorespiration. And so one of the ways that we've discovered to get around photorespiration is what they do is these plants called C4 plants, like sugarcane, which is grown in very warm tropical areas. The sugarcane, what they have figured out to do is they take in CO2 and they allow it to build up inside a special cell called a mesophyll cell. Once the CO2 is brought into the mesophyll cell, it's turned into a different compound and then transported into a bundle sheath cell. The bundle sheath cell then turns it back into CO2 and does the Calvin cycle. This way, we don't have to worry about a competition with oxygen for the rubisco enzyme. Another solution is cam plants like pineapples. What cam plants do is they only open their stomata at night. So they only take in CO2 when it's coolest, okay, when the temperature is the lowest. And by doing that, what allows is it makes it easier for that CO2 to come in and allows it to be more easily made into the Calvin cycle. In this way, we actually do the Calvin cycle at a different time. And so we don't have to worry about opening the stomata during the hottest, warmest part of the day when evaporation is more likely to happen. Now, one of the things is that photosynthesis is amazing because it controls global warming. It controls the entire climate of our globe. And that is because it limits the amount of CO2 in the atmosphere. The more CO2, the warmer the earth. And one of the things that happen, if you've ever heard of greenhouses, is what they do is they allow plants to grow all the time because they trap in solar radiation, making the temperature higher, making more CO2, so then plants can do their process better, regardless of the environment outside. However, if you have too much CO2 in the atmosphere, you are going to have global warming, and that's what we're currently seeing, is that the average temperature on Earth is increasing over time, and that's because we're cutting down trees, which take in CO2, and we're burning fossil fuels, which add CO2 to the environment. So we're putting more CO2 into the environment, we're using less of it, or plants are using less of it than ever because there's less plants than ever, and so our CO2 buildup is actually making Earth hotter. Now, again, photosynthesis plays a role in this because it removes CO2, which brings us back to our energy farms. These are farms with green plants which will remove CO2 from the atmosphere. And one of the big things that we want to worry about is how do we protect the ozone layer? And that's a quick little discussion at the back of your chapter. So here's the idea. Solar radiation or sun converts oxygen gas into ozone, which is O3. O3 shields us or protects us from UV, so it's actually a good thing. However, if we have industrial chemicals, which are called chlorofluorocarbons, we actually produce them in some of our manufacturing. It actually gets rid of our ozone. It converts O3 back into O2, which you may say, oh, that's great, there's lots of oxygen. No, it's not great because there's less ozone, which means more sunlight, which means more warmth, which means more global warming. And so that's how we can easily see the connection between photosynthesis and climate.